Hello, thank you very much for coming out on this evening. It's getting slightly colder now. It's a great pleasure tonight to introduce Professor Stephen Hillier to you, who's going to give a talk on Fertility Futures in a Changing World. Steve is our Emeritus Professor of Reproductive Endocrinology and an Honorary Professorial Fellow at the University of Edinburgh. Steve spent 20 years as Director of NHS Lothian Reproductive Medicine Laboratory, providing services in support of fertility, hormone testing and assisted conception, including IVF. Steve was also our Vice Principal International for a long time, for a period of 6 years, and developed the Edinburgh Global Brand for us. Steve retired last year in November and still maintains very good relations with the university, teaching, contributing to teaching and contributing to our research portfolio. Now, Steve's research is focused on the molecular and cellular events that regulate ovulation, with emphasis on steroid hormone physiology and pharmacology. And his research has had a real impact in the field of global health. So without any further ado, I'd like to invite Steve to come and give us a talk for this evening. Thank you.
Thank you very much, Sue. Thank you much, everyone, for coming out this evening to listen to me. As Sue mentioned, in a former life I was not just a researcher, but also, she didn't mention, I was the Vice Principal International, and as Vice Principal I was greatly privileged to work with our Changing World team that brought this series of lectures and indeed the programme into the curriculum here at Edinburgh. So it's really fantastic that I have the honour to be back in the capacity as a lecturer to actually speak to you. I think I'm right in saying that the original idea of the series, the lecture series, the programme, was to really consider the problems and the pressures that have been created in the wake of the global changes that are going on around us, and to consider how those changes might become worse in the future, and how we might, through the medium of our academic research, attempt to mitigate those changes. The list of changes, challenges, sometimes called global challenges, is quite a familiar one. I've put it up here, all the way through climate change, water, food security, energy security, etc. I think we're all happy with that list, and they're certainly the things that we focus on. But I would argue that there is one very significant omission from this list. All of these problems and pressures, challenges, are driven by the relentless growth of the human population on Earth. And population growth is intrinsically linked to fertility, human fertility. Fertility drives population growth. So I would like to argue that global fertility should be on this list and stay on this list. And in this talk I'm going to address it directly through a look at some of the changes that are occurring in global fertility, how it's actually declining now in many parts of the world. Look at how the so-called test tube baby IVF revolution that's been occurring over the last 3 or 4 decades has been and will continue to be brought to bear in tackling these problems created by the downfall in global fertility. And then share with you some rather more speculative thoughts about what may lay ahead in the future. My main messages aren't particularly startling. The first one perhaps is curious. Fertility is falling. The second one is that there will be solutions that will mitigate this through the medium of test tube baby treatment. But thirdly, the future, and forgive me for this, will be exciting because eggs will be involved and IVF will be necessary for us to move forward. And then hopefully at the end of the talk, I'll have created enough alarm, but also left enough time for us to have a discussion about some of the issues that I'm going to raise. So beginning at the beginning with a quick look at the changes that are occurring in global fertility. This is a chart from data published by the United Nations looking at the population growth that's occurred on planet Earth over the last 65 years, from 1950 to 2010 actually. And the important line there is the blue one, going all the way from 2.5 billion back in 1950 up to 7 billion in 2010. And I think this year, I believe in July of this year, or was it last year, we passed the 7.3 billion mark. So it's heading on relentlessly towards, I think the current projection now is that by the end of the century, the end of this century, it'll be over 11 billion. No surprises to see that most of that activity is occurring in the so-called less developed regions of the world, shown by the green line, and followed, albeit at a much slower rate still, in the more developed regions of the globe, shown by the red line. And of course, we class ourselves in Europe, in the United Kingdom, as part of that category within the data. No surprises here, but what I find particularly interesting as a reproductive endocrinologist, and not as a social scientist, because I'm not a social scientist, is that at the same time that population is going that way, our fertility rates are going that way. It turns out, and social scientists will be able to explain this much more clearly than I can, that as societies mature, as economic and social circumstances improve in a society, as healthcare, education, general welfare becomes more available, as people become more healthy and happy and live longer, they tend to have, indeed want to have, fewer and fewer children. So the so-called total fertility rate of a country, of a nation, will begin to decline. You can see it very obviously in the more, or rather the less developed regions of the world where this process is having a more immediate effect. Again in the green line, declining from a total fertility rate, that's number of children per woman in the population, declining dramatically down here, down to less than 3. And you can also see that even in our own societies, in more developed regions of the world, this process is still gradually occurring. What this says is, and what the projections are, is that by the end of this century, all these lines will converge, and the entire population of the world will actually be at a point where the fertility rate will not be adequate to keep the population stable at then current rates, which may or may not be a good or bad thing. I wouldn't want to conjecture. But the point is, it has massive problems for us in more developed regions of the world, because we are now at levels of population, or rather at a fertility rate, which is less than the level required to keep our population growing. And actually, if we go focus down on the European situation, we're actually moving into a situation, if not fertility failure, certainly of fertility of becoming faulty. We have, as I said, got a population with a fertility rate which is now beneath the requisite 2.1 to maintain the population. It's about 1.5 current estimates in Europe, and it's projected to stay that way at least out until 2060. And the reasons for this I've already alluded to in a broader sense, but they include our lifestyles, how long we spend going to university, what sort of jobs we have, how long we need to be in education to get those jobs, how we structure our families, how stable our jobs are, the availability, cost of housing. Generally, the entire environment in which we find ourselves normally in Western Europe, within the European Union area at least, is encouraging society as a whole to actually not want to have babies so early and so often as we used to. This is not novel, but it's important to state this as the context for what I'm going to be talking about, because by postponing pregnancy, the decision to postpone pregnancy, what unwittingly women are doing is actually increasing the likelihood that when they do want to have babies, it's going to be very difficult because they will be increasingly infertile as they age. And this is a serious problem in our society. If we look at the facts, it's reckoned that between 1 in 6 and 1 in 7 of all couples around the world are affected by infertility at some point or other in their lives. This is worldwide. And certainly these statistics are relevant in Europe. The causes of that infertility include aging, which I'll come back to in a minute, but it's generally reckoned that up to about a third of the cases are rested with the man, with male factor infertility, a similar proportion due to the female, another equivalent proportion which is possibly due to both partners, and then there is a large, somewhat smaller but nevertheless significant proportion in which the cause is unknown. Now, infertility is associated with many organic and environmental factors, but more recently we're becoming aware that lifestyle factors do have a very significant impact on our fertility. Factors such as smoking, body weight and stress. And as I mentioned already, increasing age, as highlighted here in the female partner of a couple, it is one of the most common explanations today. But the good news is, I know of hardly any reproductive condition that cannot be alleviated one way or another by the application of in vitro fertilization, test tube baby technology. Just to get it out of the way, in vitro fertilization is when you take the egg, the oocyte from a woman, you expose it to the sperm cells from a man in a culture dish outside the body, develop an embryo which can then be returned back to the woman's uterus, hopefully to grow into a baby. That technology was invented, and I'll talk about it in a minute, first used clinically 37 years ago. 1978 was the first test tube baby that was born in this country to a woman who had no fallopian tubes and was really the iconic hallmark of what test tube baby treatment can mean. And since then, we reckon that over, at least, without including what's going on in China, but where we can get data, at least 6 million test tube babies have been born since then, worldwide. And in the UK, every year, we treat about 75,000 cases of infertility with in vitro fertilization. And some 250,000 of the babies that are born to test tube treatment around the world have actually been born in the United Kingdom. And you might be interested to realize that actually over 2% of the children born annually in the UK are to test tube baby procedures. And that number is far exceeded in many other parts of the world, not least in Scandinavia, where it's reckoned now that it's headed up towards 10%. That's nearly 1 in 10 of the children born every year being born to test tube baby techniques. Well, I think that's pretty fantastic myself. And so what I would like to do now, having set that context, is to move on to look more closely at what test tube baby technology is all about and see how it's evolved as a basis for projecting changes that we might want to anticipate in the future as our world continues to change. The test tube baby technique, in vitro fertilization, which I'll now refer to as IVF, because I think there shouldn't be any confusion that IVF is test tube babies, was actually developed by these two gentlemen here, Robert Edwards, Bob Edwards, who was a physiologist who actually did his PhD here in Edinburgh at the Institute of Genetics, studying chromosomal anomalies in mouse oocytes. Bob Edwards was a physiologist who passionately believed in the possibility that IVF could occur, number one, and be adapted and used as a useful fertility technique. And Bob Edwards partnered up with the obstetrician gynecologist Patrick Steptoe, who himself had pioneered critical technologies in gynecology which allowed oocytes, eggs, to be collected safely from a woman's ovary so that the IVF procedure could be conducted. I just want to elaborate on or emphasize the fact that we should be celebrating Bob Edwards' association with Edinburgh, because he was a truly remarkable man who brought to bear really this entire process. He actually was given a bursary by the University of Edinburgh of the princely sum of £240 a year whilst he was here as a PhD student. That's back in the early 1950s. And oh, if we had known that he would eventually become a Nobel Prize winner in physiology or medicine as he was in 2010, I think we might have looked after him a little bit better whilst he was here. But that's the way it stands. It was through the partnering of this physiologist, Bob Edwards, with the obstetrician gynecologist that we were able to see the advent of IVF procedures. This image here is from the front cover of a memoir that Edwards and Steptoe published in 1980. And I thought that by just opening that book would be the easiest way to give you a sense of what was accomplished leading up to the advent of the first test tube baby. Actually, this is an image from one of the chapters, which is a montage of all the critical steps in the development of an egg and an embryo that are required to occur in vitro if in vitro fertilization is going to work. And all of these images were recorded, obtained and published in various papers mainly in Nature during the 60s and early 70s, leading up to the first clinical application of IVF. The image of an oocyte waiting to be fertilized, the image of a sperm cell approaching the surface of an egg. None of these things have been seen so clearly before. An image of the sperm cell actually penetrating and associating with the outer membrane of the oocyte. An image showing fertilization actually occurring. The smaller female egg nucleus, the larger, always larger male nucleus from the sperm. Then subsequent cleavage of the embryo, a 4-cell embryo. Of course it goes 1 cell, 2-cell, 4-cell, 8-cell, 16-cell, 32-cell, 64, until we reach the advanced stages of development which precede the formation of the blastocyst. The blastocyst showing the embryonic disc that will become the baby once there is implantation, and the outer trophectoderm layer of cells which will become the placenta. All of this done before the technique was finally developed. And before now bringing you up to date with how modern IVF is being done, I want to just give you a quick primer on the natural process of reproduction in human beings. I'm sure you'll say, well, why are you going to do this? But I'm assuming that some people here are social scientists or engineers or other from other specialities that won't have quite such a nuanced understanding of reproductive physiology. This is just a simple diagram of the uterus, of a woman's uterus, cut or exposed to show the inner cavity, which is connected to the vagina via the cervix. And at the top of the uterus on either side is the so-called fallopian tube that leads out. And at the end of each fallopian tube, there are the so-called fimbria, finger-like structures that stay in the vicinity of the ovary, waiting for an egg to be produced that will be taken into this apparatus. So what happens is, first of all, we get ovulation, and an egg is produced. Secondly, we get insemination, and sperm are produced. It doesn't actually have to go around that way, because sperm survive in the reproductive tract for quite a long time, in fact up to about 5 days. So these sperm cells can actually be laying in wait for the egg as it's picked up and conveyed back into the tube. But either way, both ovulation and insemination have to occur so that fertilization can occur. Now the big trick about this technology when it was first introduced in 1978 is that for the first time, women who didn't have normal fallopian tubes, or even didn't have fallopian tubes, because maybe surgically they'd had to be removed in the past, or they'd had intra-abdominal disease or burst appendix or something which had caused their tubes to be damaged. Women in this category without patent fallopian tubes could not have babies, except possibly in some cases by tubal microsurgery to open the tubes, but that was extremely rare and in itself relatively unsuccessful. So IVF, to avoid this problem, was a massive advance. But to continue the primer, so the sperm and the egg have met each other within the fallopian tube. Fertilization occurs, and embryo cleavage then begins. 2-cell, 4-cell, 8-cell, 16, as I showed you, all the way up to the blastocyst, which then is ready to implant into the wall of the uterus and start the pregnancy. That's the normal process of events, and all IVF does is to circumvent a large proportion of those steps by doing it in a dish. Actually, it's always been in a plastic dish, not a glass dish, in vitro, in glass, but it's done in a dish. The entire process of fertilization, cleavage, to the point of obtaining an embryo that can be implanted into the patient's uterus. Let's look then at the processes that now pertain, if we're going to do this real time now, 2015. In order to do IVF, we need first of all to get eggs, and that's done normally by stimulating the ovaries with fertility drugs, gonadotropins, which enhance the number of follicles that grow in the ovary. This is a visualization done by ultrasonic imaging. Enhance the number of follicles present in the ovary containing mature eggs that we can collect to fertilize to turn into embryos. And to get those eggs out of the follicle, we need to stick a needle into those follicles, aspirate the contents. Again, this is done by ultrasonic guidance these days, but this is a nice old photograph actually looking into the abdomen at the time that eggs are being collected, showing all the follicles that are present, each of which will be sucked out one by one using special evacuation equipment to draw the fluid into a test tube, which then goes to the laboratory where the scientists involved in the process, the embryologist, will search for the egg. Here's one there. It's very difficult if you don't know what you're looking for. It's the largest cell in the body, but it's amazingly difficult to see sometimes. And if I highlight it there, there it is. That's the egg surrounded by a compact layer of granulosa cells. In an ideal scenario, there will be up to about 10 oocytes, eggs, collected. And then the number of eggs collected normally determines the number of embryos obtained. And that normally determines the likelihood of success, because it means that there are plenty of embryos to be used to attempt to obtain a pregnancy. But in the laboratory, the embryologists will be taking the sperm, which has already been collected from the male partner, and prepared for mixing with the oocyte so that fertilization can occur in vitro. And if and when fertilization occurs, we can recognize it, as I showed you from the Edwards images that he had observed, the two nuclei, the smaller female and the larger male nucleus, which will prove to us that fertilization has occurred. Actually, many, if not most, forms of infertility are due to problems with sperm and with the man. And there was a true breakthrough in vitro fertilization about 20 years ago, when a new technique was established, which meant that you didn't have to fertilize eggs just by mixing them with sperm and hoping that sperm could fight their way into the egg. You could actually inject the sperm cells straight in. A single sperm cell using micro-manipulation technique could be injected directly into the oocyte, as shown here, and as shown on this film. I'll show it. It was kindly given to me by Dr. Sue Pickering, who is the senior scientist at the in vitro fertilization unit here in Edinburgh, the assisted conception unit. And it shows what happens. I don't know if you saw the sperm, it's still in this needle. It was in ICSI, the idea is that you take the sperm into the oocyte through this needle, take back a little bit of ooplasm to mix the oocyte into the ooplasm, and then put it back into the heart of the egg. And now that egg has been set up to fertilize. That sperm cell could have been completely immotile, it could have been completely immature, but fertilization can still now occur. And actually, up to 60% of all fertilization procedures that are done in IVF in this country, even if the sperm is completely normal, are now done using this procedure because it's so successful. Anyway, once we've got a fertilized egg, either this way or that way, we will then expect to see the cleavage steps occur 2, 4, 6, 8, and so on, as I said before, all the way up to here, hatching a blastocyst ready to implant into the wall of the uterus about 5 days after fertilization. We want as many of these as we can get, as many healthy embryos as possible, and then attempt by attempt they will be replaced into the woman's uterus. This is the business end of an embryo transfer catheter, magnified several hundred times. The idea is that this catheter will be loaded up with an embryo. Here's an image here. And then the catheter will be threaded up through the cervix into the uterus, and with a syringe applying positive pressure deposited in a droplet of culture medium into the uterus. Just like that. That's not good enough, is it? Let's look at it properly. Here again is another image from Sue Pickering that actually shows us, what's going on. Let's try and image it for you here. Can you see this catheter being threaded into the uterus, and then poof, you'll see a white, there you are, that's the embryo going in. I don't think embryos make that, expose themselves in that way, but I think there's a little bit of air that was behind the embryo that would have gone in at the same time, very conveniently. It's common practice now to visualize transfers, ultrasonic exactly like this, and I know that patients in particular like to see this actually happening and to be reassured that their embryos have in fact gone back safely as part of the procedure. So again, I thank Sue for letting me show that image. Once the embryo, we wouldn't transfer normally more than 2 embryos nowadays. We used to transfer many more, but nowadays it's common practice to replace only 1 or 2 embryos. Once the embryo has been replaced, we would expect attachment to start to occur about 7 to 10 days later. And then the embryo, as it grows, as the placenta forms, as the pregnancy develops, will start to transit these classic stages of embryo development, all the way up to the stage at which it becomes what we would call a fetus rather than an embryo, which is at about 60 days post ovulation or post-fertilization. It's important to emphasize that not all embryos make this journey, and of course there is a relatively high attrition rate, and as many as 30% of the embryos will typically not proceed due to this entire developmental stage due to chromosomal anomalies. And there'll be other reasons why patients will also not become pregnant. I do not want you to go away with the idea that these techniques are always successful, but they do occur with a high degree of success. And it's not unusual now for a patient, depending on age, reason for infertility, etc., to have literally a 70% likelihood that she will become pregnant from the batch of eggs that are collected from an oocyte retrieval attempt. Bottom line is that this is now commonplace procedure. It's very widespread, it's very safe, and it's very effective. And it does far more than simply treat tubal disease, as I introduced at the beginning. Only some 9% or so of cases are actually of that etiology. There are many other reasons: ovulation problems, endometriosis, multiple factors. Male factor itself is a massive part of the problem with fertility. Then there's a whole slew of unexplained reasons for infertility, as well as multiple male and female factors, all of which can be successfully treated by in vitro fertilization. And it's very helpful for us in the area to be able to call on the data like this that are published by the so-called Human Fertilization and Embryology Authority, because as you'll see, this particular pie chart comes from the UK census in 2013, published by the HFEA, of all of the treatments of women that were conducted using their own eggs by IVF. And this is a good prompt for me to introduce to you the Human Fertilization and Embryology Authority and highlight the importance that the authority's work has on the development of the field of assisted reproduction, and IVF in particular. The Human Fertilization and Embryology Act of Parliament was enacted in 1990 in the wake of the huge amount of controversy that surrounded the development and introduction of the IVF procedure. And part of the Act's purpose was to create the so-called HFEA, Human Fertilization and Embryology Authority, which was mandated with the responsibility to regulate everything to do with human embryo research, to license that research, to regulate and license any treatment that involved the use of gametes, eggs or sperm or embryos outside the body in vitro, and to regulate all procedures around the donation and storage of eggs, sperm and embryos. And also, last but not least, to maintain a registry of all the procedures and outcomes that occur. And as I showed you before, there are some 75,000 IVF cycles a year that go on in this country. So that's a huge amount of important data and regulatory activity that goes on. Subsequent to that Act of Parliament, there was a need to quickly introduce another act when Dolly the sheep came on the scene, and there was a worry that cloning and related procedures would not be adequately covered by the original Act of Parliament. But then a few years later, both of these acts were rolled into Awana, and the HFEA Act 2008 continues to ensure that the entire field of assisted conception, the research around that area, the treatments around that area using human embryos and gametes is closely and tightly but sensibly regulated. And it's important to emphasize how privileged we are in the UK to have such an effective body, and how much good work, I would argue, they are doing. The UK was the first country in the world to produce test tube babies. It was the first country in the world to introduce regulation of all the procedures around the production of test tube babies, and it continues to be a bastion of best practice in keeping eyes and ears out and open for changes that occur in the field. So as the test tube baby explosion has emerged, the authority has been there right in the thick of it, overseeing, encouraging, and if you like, making so transparent and accessible all the new developments that are occurring in the field, to scientists, to clinicians, to patients, to the lay press, etc. Over those years, there have been huge challenges due to the new scientific developments that have continually emerged. We all know about Dolly and stem cell nuclear transplantation as a cloning technology. That caused a huge stir at the time. Then that problem morphed into stem cells, stem cell biology, the way in which they might be used, which morphed into our ability to make synthetic eggs, synthetic sperms, using stem cell technologies, which morphed into problems to do with mitochondrial donation, which morphed into genome editing, and so on and so forth. There's always something out there testing and challenging and creating new controversies that we all as a society and the authority as an agency have to deal with. And of course in their wake are the social challenges that have been with us and are still with us as the field marches on. I mentioned 3 here. I'm going to resist, however, the temptation to dwell on donor conception. Suffice to say, it is an ongoing issue to do with sperm availability, issues around donors, issues around where the sperm comes from, and issues around how long it can be stored and how safe it is, etc. I also resist the temptation to dwell too much on surrogacy, another very contentious and controversial issue which we may want to come back to in a minute. But I would like to dwell now specifically on the issue of older mothers created by the tendency to postpone pregnancy and thereby increase infertility in our society, in other parts of the so-called developed or more developed regions of the world. These particular results here, our data here, are just a summary of changes in live births in the UK, actually not UK, sorry, England and Wales. These are data published by the Office of National Statistics. Over the last 2 decades, characterized according to age groups, less than 25 to 34, greater than 34. And the point to notice really is that the number of first-time mothers in the UK, in England and Wales, over the last 20 years, after these last 2 decades, the number of first-time mothers under the age of 25 was almost halved. And over the same period of time, there's been a tripling in the number of first-time mothers over the age of 34. This is in the population at large, but this illustrates the changes that are occurring when we talk about delayed or postponed pregnancy and so-called older mothers. And it puts, again, the context to this data here, which shows how the age of women submitting to test tube baby procedures has increased so strongly over a similar period of time. Looking at the proportion of different women of different age groups who went in for IVF back in 1992, when the HFEA first started to do its work collecting data, as compared with recent data in 2010, you can see this massive right shift where we've seen a move from a peak of activity in women at around the age of 33 off to here in 2010 going up as high as 40. And from there onwards, at least 50% of all the women presenting for IVF in the country are older than 35. And that is quite a profound statistic when you consider what the consequence of that is for their likelihood of treatment success. Because here you are looking at IVF pregnancy rates in relation to age from 2 years of data from the HFEA, just because it emphasizes the point that this is a non-changing phenomenon, that women under the age of 34 have a relatively high likelihood of pregnancy success, but as they age, that likelihood of success genuinely crashes, and we get to the point where around the age of the early to mid 40s, it really becomes a heroic procedure to try and get such women pregnant without resorting to the use of donor oocytes. And that's another issue of controversy that we might want to discuss in a minute. Suffice to say that this is something that I think we're all becoming aware of. The media are constantly reminding us of it. Certainly mums are constantly reminding their daughters, if the media aren't constantly reminding them, that they better get on quick if they want to have babies, because sooner or later their options are going to run out. That's simply because their eggs are disappearing. As we sit here, as we stand here, our eggs are disappearing. Sorry, but it's true. And here's an example, real time. This is the cortex of an ovary from an 11-year-old girl, showing eggs, 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 eggs everywhere. This is the cortex from a 34-year-old woman who had a piece of her ovary preserved. This is 34. This is good news. This is supposed to be a good time to try and get pregnant. But even there's hardly anything. So it just shows you in a wonder exactly what's happening with age. Eggs disappear. And they decline in quality, are less likely to fertilize, less likely to develop into embryos. And the embryos that develop are less likely to develop into babies. And because of this, it's become quite common, as I say, to advise people, encourage people to consider either getting pregnant early in their lives or taking measures to avoid these problems, such as storing their eggs. It is technically possible now for a young woman to have her eggs collected and frozen, cryo-stored for future use. And apparently, there are even companies, Apple and Facebook, allegedly, that offer this as a perk to their patients so that they can actually get on with their jobs rather than waste time getting pregnant, having families. Another issue for discussion. All in all, the whole field has become a complete global business. It's reckoned that there is something like what does it say, 10 billion market, US dollar market out there that's going to grow to over 20 billion over the next 10 years. And you can calculate that every test tube baby that's produced in Europe costs about 35,000 euros to produce. This is a big business as well as an important medical advance. And it's an area that I would suggest, if you've got any spare cash, is worth investing in at the moment. Okay, future scenarios. This is where I start to speculate. I was told to be provocative, so I'll do my best. Going back to Bob Edwards, who sadly passed away in 2013, even before the first test tube baby, just 10 years after he had got his PhD from Edinburgh, he published this paper describing for the first time ever the observation of human eggs maturing in culture. Look how touchingly he advertises his Edinburgh connection there. Even before Louise Brown, the first test tube baby, was born, the procedure had been validated. He was looking ahead, not just of the time at which in vitro fertilization could be used to treat infertility, but beyond that, to the time at which even control of genetic disorders in patients or embryos might in fact be tractable by in vitro fertilization. This is 50 years ago, almost to the day that this paper was published, way ahead of his time. Edwards himself was actually a pioneer of genetic diagnosis. He was first described the sexing of rabbit embryos using cytogenetic procedures. So he knew the power of these procedures. But what he was never able to accomplish, it was before his generation, was was genetic diagnosis on living embryos. And it wasn't until 1990 that was first ever accomplished by the group led by Robert Winston, Lord Winston, at the Hammersmith Hospital. The scientist involved was Alan Handyside principally, when they were able to demonstrate that by taking a single cell from a human embryo, they could genetically diagnose its predisposition. In this case, they sexed the embryos to recognize Y chromosome-bearing male embryos, which would not be transferred to the patient's uterus because they would be likely to show the phenotype of X-linked genetic disease. This was a crude first attempt at what became known as pre-implantation genetic diagnosis. And with the publishing of this paper, Pandora's box was truly opened. This procedure is now, if you like, the gateway to the future that I'm now going to talk about. Because in its simplest form, it's all about using those micromanipulation procedures to take a single cell from a growing embryo made in vitro, test it with what now are some of the most advanced, precise, efficient and effective genetic screening techniques that we've ever had and which are improving on a daily basis. Select embryos that pass or fail that genetic test, or reject embryos that pass or fail those tests. But either way, put one of those chosen embryos back into the uterus to develop into a baby. It's now possible to create a genetic barcode for an embryo, an entire human genome, before it's put into the uterus, which opens up huge possibilities that we might want to discuss. In the UK alone, it's reckoned that well over 400 genetic traits have been treated, avoided in this way, using PGD. And it's also possible to do HLA typing, search for chromosomal abnormalities to avoid putting abnormal embryos back into the uterus, as well as sexing for X-linked diseases, as I just mentioned. Note, this is for medical reasons, not for social reasons. Selection of male and female embryos, although it's very easy, is illegal for anything other than medical purposes. It is not allowed in this country, although it is in America, for example, for family balancing. But either way, the advent of this procedure has changed the whole landscape of IVF. And it's the first example, as I cruise into the end of the talk, of some contentious areas that have evolved in the field of IVF, and they're going to be there as problems for us in the future. Let's look at egg banking. I already mentioned the advice and the expectation that that some women are receiving regarding banking of eggs. Yeah, we can bank eggs. We can do it now safely and efficiently, but it's a relatively new proposition. Only in recent years has it been possible to cryo-store eggs. I told you they're the largest cell in the body. That makes them the most difficult cell in the body to freeze without destroying. But developments in low temperature biology, as it's known, have resulted in the application of vitrification to oocyte cryostoring, which is much more successful. So it is now genuinely feasible for women to cryostore their oocytes, should they so wish. And as I've already mentioned, or perhaps I didn't, it's now the case that over half of women over 45 are having their IVF treatments using donor eggs. So cryobanking is a real part of the present and will become more so in the future. I also nominate robotic IVF. IVF is an incredibly fraught, complicated, dexterous procedure done by human beings. Humans have to do all the handling, make all the decisions, and implement the recommendations. This is both the scientists and the doctors and the nurses, everyone involved in the process. Wouldn't it be good if it could all be done robotically? Well, it can, much of it can. And there are systems available now where embryos can be fertilized, cultured in culture media that are constantly being adjusted in terms of composition and osmolarity, etc., in order to maintain the appropriate environment for fertilization, for early cleavage to the 2-cell, for subsequent cleavage to the 4-cell, and so on. The whole process can be effectively done in, if you like, a robotic manner. There are systems available, and it's almost inevitable that they will be introduced into practice as they improve. And the only problem there is, of course, that some embryologists at least are likely to be out of a job. The other thing that's massively changed and will continue to change the field is embryo imaging. It's now possible, as I'll show you here, again this is one of Sue's donations to my talk, to actually watch an embryo undergo fertilization, or rather cleavage at least, in vitro, as I'll show you with this image here. There are now culture systems which allow you to culture up to 72 embryos in the same piece of kit, and each one of them be constantly monitored by a camera all the way through the 2, 3, 4 days during which fertilization, cleavage, up to the stage of a morula, in a minute a blastocyst, at the time of which it will be replaced back to the uterus. This can be monitored faithfully, real time. Embryologists always believed that the speed and symmetry with which cleavage occurred was a good index of embryo quality. But they could never really prove that, because every time you looked at an embryo, you had to open the door of the incubator, get a microscope and look at the embryo, and each time you did that, you were really exposing the embryo to very adverse conditions, changes in temperature, exposure to light, etc. But now we can see it very, very clearly, because this is, for example, we know an embryo which developed into a baby. And you may have noticed as that cleavage occurred, it was impeccably symmetrical. 1 went to 2, went to 4, went to 8, and so on. And a good embryologist will be able to look at these records and be able to pick out exactly which is the best embryo to replace to maximize the chance of pregnancy occurring. Another thing that we have to worry about, or rather not worry about, but consider as we go on, is the impact of culture media on the outcome of the procedure. Just because you have a baby born through the procedure doesn't actually mean that it hasn't been affected, possibly adversely, by that procedure. You'll all have heard of the so-called Barker hypothesis, which says that the external environment and indeed the intrauterine environment during gestation can impact the health of a child and subsequently an adult. Exposure during uterine life to adverse circumstances, stress, poor nutrition, can pre-program a tendency to disease. Well, here's an example of it happening real-time in vitro in a culture dish. Because from this Dutch study that was just recently published, you can see that babies that were born in this culture from embryos that were grown in this culture medium were consistently, only slightly, but consistently larger than those born in this other culture medium. You might say, is that important? I don't know. Should we worry about that? I don't know. Does it make a difference? I don't know. But there is a statistical difference in this parameter, and it does suggest that we are potentially adversely affecting the future of an individual through not optimizing the environment in which culture is occurring. I've already mentioned the new developments in gene sequencing. They have revolutionized embryo diagnostics. Over the period since the human genome sequence was concluded back in, what was it, 2003, we've seen a decline in the cost of getting a single human genome fall from something about $100 million, which was the cost of the Human Genome Project, to almost $1,000 and soon less than $1,000. And over the same period of time, the technology has so massively improved that the speed with which the sequencing can be done means it's possible to collect, determine an entire genome within a matter of a few days, if you've got the technology at your disposal and if you've got the bioinformatics facility to use the data you can collect. Point is, there has been a complete sea change in diagnostics around the use of gene sequencing, and that opens even more avenues for future research and for discussion perhaps later here. Because finally, I talk about the possibility of gene or genome editing. It's been the nightmare of society that one day someone's going to genetically modify an embryo or clone an embryo or do something that would fulfill society's worst expectations around the slippery slope that we're on through test tube baby procedures. It's not been legal, it's not been feasible, it's not been safe, it's not really been on the agenda as a practicable procedure. Anyway, it's illegal, and anyway, why would we want to do it? But the point is that there are now new technologies available to us which make it very, very easy to do. And in the last year or so, there is a new technology, so-called CRISPR-Cas9, which many of you here will understand more than I do, but it's a technique which allows the editing DNA enzyme complex to be injected into a fertilized egg, male, female, you've seen this, male, female nucleus, and to develop an embryo which, when placed back into a mother, will have selectively targeted, modified genetic changes. The proof of concept is there. It's been done in monkeys. And these are the guys that had it done to them. Genes can be modified in primates. Can we do it? Would we do it? Why would we do it? How would we do it in human beings? Don't know. But already there is the first publication from a group in China demonstrating an attempt very responsibly to validate, in no sense to try and achieve embryos for treatment purposes, but to validate the possibility that the technique might work with human embryos. Turns out that their data, they were using already damaged oocytes that hadn't fertilized properly. Turns out that the procedure is far from as safe and selective as has been mooted and as is predicted on the basis of animal experimentation. But the point is, we are now in that domain, and we really do have to watch this space. So my conclusion is that in the next generation of IVF, there will be massive changes involving, at the very least, gamete banking and robotic procedures, which will improve treatment options and treatment outcomes. And yes, we will also see much more intensive monitoring. I showed you the real-time imaging, also the genetic programming or oocyte culture medium programming. We have to be aware of this, that the environment in which we are growing embryos will have to be always maintained optimally if we want to make sure we have healthy embryos and healthy babies. Yes, you can anticipate far more extensive and intrusive gene screening technology to ensure that we select the healthiest embryos for development. Possibly, who knows, we will see genome editing being introduced in some form or other to treat genetic disease, if not to Vobitidas modify genetic traits. What does this all remind us of? You've all read this book, and I had to resort to it. It's a cliche, but it's there. You've got to read it again if you haven't read it for a while, because in the Brave New World, there are hatcheries where gamete banking and robotic procedures occur. There is a whole section of the Institute of Hatcheries and Conditioning, where embryos are cultured literally under environments that are designed to affect their subsequent development. There's a whole issue around predestination of those embryos that are in culture in terms of what sector of society they're going to fulfill. Are they going to be alphas, betas, etc.? And then there's a whole issue around enhancing individuals so that they grow into the sector of society that they have been predestined to occupy. Fascinating stuff, which you might say, but for goodness sake, that is overly sensational and perhaps inappropriate in the context of an intellectual discussion like this. But I think not. I would point you to a recently published document published by the Ministry of Defence, the UK Ministry of Defence, looking at issues that challenge our society now in terms of our defence and future economic viability, and how we should gear ourselves up to anticipate and protect ourselves to or against, prepare ourselves for those changes. It's a hell of a read. I don't know if you've come across it yourselves, but it's over 170 pages long and it's dense. But right at the end of the section on medicine, there are these words. You can read them yourself. Antimicrobial resistant infection could significantly increase medical risk on military operations. Yeah, okay. Novel medical and surgical interventions will almost certainly improve casualties, survival and recovery rates. Yep, good. Advances that allow patients to interact with their prosthetics and other aids are likely to lead to new ways to connect the able-bodied to machines and computers. Interesting, optimistic, scary, feasible. But last but not least, some countries and individuals are likely to use advanced medical techniques such as genetic modification to gain a competitive advantage. Others will probably constrain their development for ethical reasons. I rest my case there and wonder which camp we will be in. Now we can have a discussion. Thank you.
Thank you. I'm really enjoying about it. I'd like to know that is the frozen egg as safe as before if used after 10 years?
Could you just say that a bit?
Is the freezing eggs as safe as before if I used after 10 years?
Freezing now is much more effective and safer because of the introduction of the procedure called vitrification, which means that ice crystals aren't actually formed in the oocyte, or less likely to be, well, they're not formed in the oocyte, which means that the lethal effect of ice crystals on the chromosomal structures within the ovary, within the egg, are not there. So egg freezing now is much, much safer than it used to be.
Does artificial insemination damage the egg?
Where are we? Sorry. Artificial insemination, no, not at all.
You're sticking a needle through the wall, though.
Oh, sorry. ICSI, I beg your pardon, because artificial insemination also means insemination of a woman with sperm naturally. Does it damage the egg? There has been a huge controversy around this, and for years there were worries not so much that pricking it with the needle would damage the egg, but that where the needle went inside the egg could cause further damage, for example, to the structure of the chromosomal apparatus, the mitotic spindle, for example. And it can cause damage. But there are very tight protocols that are used by embryologists to do this procedure, which minimize the likelihood of that damage. And as far as we know, after a couple of decades of experience now, there is no evidence of any particular damage using this sort of technique. But it's always something that one has to bear in mind, and who knows, could show up in the future.
You said such a high price per each IVF baby in the EU, about 35 grand. I'm wondering what area would you see as room for where that price would drop? Like which, I suppose, do you think that the price will drop?
Yeah, I mean, there is actually an emerging trend or binary here of high-tech and low-tech IVF. A lot of the future I was looking at there is predicated on high-tech, where we are now. But a lot of the costs of IVF can be mitigated by using relatively simple procedures, by minimizing the amounts and type of fertility drug that's being used, because that's a huge expense in its own right. And also by actually designing customized fit-for-purpose facilities that do just IVF and do it very effectively. So there are definitely areas where, and there have been examples of where improvements and reductions in cost can occur. And you have to bear in mind that the real issue here is that most IVF treatment in this country and many other countries is done in the private sector, and the fees that are charged to the patient are consistent with receiving private treatment, and that really can add substantially to the cost of the treatment.
Thank you for a very interesting talk. Given developments you've shown in IVF and separately in tandem developments in neonatal care, how far away are we from the Brave New World hatcheries where a whole baby could develop in vitro?
Yeah, no, I searched for some massively over-sensational example of possible babies being grown outside the body, etc. It's quite interesting that there are hints around sort of fetal physiology in sheep models and in domestic animals in particular, that certain quite advanced stages of fetal development can occur, albeit suboptimally, in vitro. And there are all sorts of wacky things like, in theory it's possible for a man to carry a baby to term if the appropriate hormones and so forth are given. So therefore you could envisage it being done in surrogate animals or something like that. I mean, you can get quite bizarrely engrossed in the possibilities if you follow the line of thought ad nauseum. But there is nothing scientifically or medically convincing to suggest that's anywhere near likely. There are, though, of course, as you'll be aware, then, recent examples of uterine transplants where women who do not have a uterus, of congenital or for surgical reasons, have had a uterus uterine transplant, and managed to carry a baby to term in Sweden, Gothenburg. So, if you put everything all together and look at the animal evidence, the advances, small advances that have been made clinically, and the technical feasibility that is now there in front of us, staring at us, of so many biological processes being adapted to other purposes, then you'd have to believe that sometime, somewhere down the line, that would be feasible.
So I was just wondering, so it seems that one solution is to get pregnant quickly. The other solution is to freeze the eggs. And I was wondering, is there any research on any possible interventions that would slow down the rate at which females are losing the eggs?
The answer is yes. How can I address this easily? What I didn't explain is that it's a natural process. Well, I did, but in a rather flippant way. It is a natural process for the oocytes, the eggs in the ovaries, to gradually disappear throughout life until the menopause is reached. That's effectively what causes the menopause, the loss of eggs. And the process whereby that occurs is known as programmed cell death. It involves apoptosis, which is literally the involution of those cells with time. And it's a genetically regulated process. There are pro and anti-apoptotic factors which set the speed at which that process occurs. And in mouse models, there have been attempts, to a greater or lesser extent successful, to try and manipulate the expression of those genes to regulate that process. And certainly it's feasible that using that kind of technology, it ought to be possible to slow down the rate of atresia of those oocytes, and hence to delay that process. But to see that as a clinically viable option, I think would not be realistic at the moment. But again, it's something which, if it can be done in an animal somewhere, or a fruit fly, or a yeast cell, or you can almost be certain that it would be feasible in higher organisms as well. Whether or not we would want to do it, whether or not it would be safe, whether or not it would answer these questions or these expectations, who knows? That would be another controversy of another era.
Thank you for your talk. I was wondering if you could expand a little bit, if you know any more, about that statistically significant difference in the embryos, depending on which medium they had been.
Yeah. Well, this is a real hot potato, Because for years, right from the very beginning when we first started, when I used to do IVF, we used to make our own culture medium, you know, with a scale, you know, sort of salt and some colours and some sugar and a few vitamins and everything, and then off you go. But the good thing about that was that we knew exactly what was in it. We could measure what was in it. We could adjust what was in it. And it may have been quasi-optimal as it happens anyway. But what happens now is that IVF clinics wouldn't be licensed to function unless they bought a BP equivalent, you know, pharmacopoeia, medical safety ratified quality, medical grade culture medium. And actually, those culture media, although up until recently we didn't even know what was in them. Now we know what's in them, but they don't tell us how much of whatever is in them. And anyway, you can't control it once it's there. And so the whole field is being conducted using commercially procured culture media. Both of those that I showed there are commercial. And it's a real problem. That difference was statistically significant. And it has been repeated. But I wouldn't, again, I would sort of try and emphasize the fact that even though there is a difference, it doesn't necessarily mean it's an important difference or it's going to have any sort of meaningful effect on quality of life, so to speak, of those children. But you have to ask yourself that the question, what culture medium was I conceived and grown in as a test tube baby, might be one that you would want to have answered at some point. And it's certainly something that's being looked at very closely now. Thank you.